Hello, my name is Judith Klein-Seetharaman from the Department of Chemistry at the Colorado School of Mines. I will be talking about our pipeline for discovery of membrane receptors in non-model organisms, the case of Pocillopora damicornis. Frequently we hear about corals in the news. For example, it was recently reported that the Great Barrier Reef suffered the worst ever coral bleaching event this year. What can we do about this as computational biologists? Our hope is that by harnessing the data revolution, we can come up with ideas on how to save the corals. We have an enormous opportunity provided by the availability of large data, especially full genomes. But the challenge is, how do we interpret this massive amount of data? Here we want to leverage well-studied organisms, such as human, to interpret the coral genomic information. In the long term, we wish to use this information to design intervention approaches to prevent bleaching or to treat bleached corals. Our approach uses remote homology detection and analysis. Why remote? Well, the corals have evolved before the split into deuterostomia and protostomia. Deuterostomia is where humans are, and protostomia is where Drosophila and C. elegans are. That's a long time for mutations to accumulate, so we expect many homologs between human and corals to be in the gray zone of about 20 to 30% sequence identity at best. The coral we have chosen for this project is Pocillopora damicornis, a stony coral that makes its own calcium carbonate skeleton and houses colonies of individual animals, the polyps, which you can just barely see here by eye. Once magnified, you can see the individual corals very well. The holobiont is comprised of the cnidarian animal host, as well as a microbiome of over 20,000 different species, from the symbiotic algae to bacteria, fungi, and viruses. So what do we know about coral behavior in response to environmental cues? We know that corals manage 90% of their energy from symbiotic algae, which means they can measure and regulate the nutrient balance. Corals can see, they can detect and respond to light. See, for example, the image here of a xenia coral in the dark, all huddled up, and in the light, all spread out. Corals can fight, they can sense and attack their enemies. Corals can tell friend from foe in symbiosis versus immune attack. And corals prefer to eat plastic over copepods, which is somewhat questionable, but not so much when you think about humans eating refined sugar rather than salads. So how and why do the corals do all these behaviors? And maybe can they do other things that we don't know about yet? To address this question, the next question is, well, how do humans sense and respond to environmental cues? The answer is through membrane receptors. The most important class of membrane receptors are the so-called G-protein coupled receptors, or GPCRs. GPCRs are 7 transmembrane helical proteins which bind a very large diversity of different types of ligands, all the way from small molecules to entire proteins. They bind to the 7 transmembrane helical bundle in the extracellular domain and cause a conformational change that results in binding and activation of the G protein and ensuing signaling cascades. The GPCR family is the pharmaceutically most important protein family in humans, with more than 50% of drug targets targeted at GPCRs. In addition to GPCRs, which are also referred to as type 2 membrane receptors, there is the so-called type 1 receptors, which have only 1 or 2 transmembrane helices and are coupled to enzymatic activity in the cytoplasmic domain. Most common families here are the tyrosine kinases and phosphatases. In order to identify which sensing and signaling capabilities the corals have, we've taken the following approach. Step 1, identify all membrane receptors in human. Step 2, map these receptors to Pocillopora damicornis, which I'm going to refer to for the remainder of this talk as PDAM. Then identify what other proteins are in their respective signaling pathways, and then derive hypotheses from this. We will now describe step 1. These are the sources of membrane receptors that we have used, ranging from 825 GPCR proteins in this list to 978 proteins from 2003, which has been updated to include about 1,300 proteins in 2009. We then map these receptors, sorry about that, to PDAM proteins by using a method called HH-Blitz. HH-Blitz is a hidden Markov-based model that allows retrieval of remote homologs using an HMM to HMM alignment. This is a unique aspect of this method where the query sequence itself is converted into an HMM by using these amino acid distributions shown on the right-hand side, which results in really very exquisite sensitivity of this method, shown here by their 2011 paper in demonstrating how many more remote homologs HH Blitz find compared to traditional profile HMMs. And we can confirm this. For example, you can see here an excerpt of the alignment of toll-like receptors where there's very high confidence in the alignment. And if you look at these individual residues, they totally make sense. And we were not able to obtain this kind of an alignment with profile HMM-based methods. And so we are fortunate that our collaborator at Tufts University, Professor Lenore Cohen, and her graduate student Montserrat have developed this interface for us, which makes it particularly easy for non-computer scientists to use HH Blitz. Applying HH Blitz then to the 3 membrane receptors resulted in about 330 PDAM proteins, out of which 150 are predicted to be GPCRs. This is roughly a similar distribution compared to human membrane receptors, where also about half are GPCRs versus non-GPCRs. So next, we looked at what are the proteins in the signaling cascades. And we've used the insulin receptor signaling pathway as an example. So we've retrieved all the PDAM homologs for the insulin receptor pathway, but we've also looked at 2 other pathways, the glucagon signaling pathway and the somatostatin signaling pathway, because these are known to be negative regulators of insulin signaling. Everything shown in black here means that there is PDAM homologs available. Everything shown in red is that we couldn't find a PDAM homolog. You can see that there's a lot of crosstalk between these pathways. For example, here is proteins that are actually represented in all 3 pathways, some of which are only shared between 2 pathways. And so you can see that there is ways to communicate across these pathways. What's particularly interesting to us is that in the insulin receptor signaling pathway, only 1 protein is missing, namely BAT, which is a downstream factor that leads to upper process. But in contrast, in these negative regulators of insulin signaling, we don't find the ligands. We are missing the glucagon and we are missing the somatostatin. It's very tempting to speculate that corals can regulate nutrient uptake and release via insulin signaling, but that the regulation of insulin signaling by other pathways may not actually function. We speculate that perhaps these ligands may be provided by the microbiome. Maybe bacteria, algae generate ligands to negatively regulate insulin signaling to keep the nutrients for their own use rather than allowing consumption by the corals. This brings us to the final pipeline step, of course, deriving biological hypotheses. And so we are going to demonstrate this using a GPCR example, namely the prototypical and name-giving class A GPCR rhodopsin. This is the picture of the structure of rhodopsin, and you can see its retinal ligand binding pocket. This is a vitamin A derivative shown here. Upon light, the 11 cis-retinal isomerizes to all trans-retinal, which causes a conformational change in the transmembrane domain that's transmitted to the cytoplasmic domain, where it induces the binding and activation of the G protein, phosphorylation by a rhodopsin kinase, and ultimately binding of arrestin, which shuts off the signal. And so what you can see here is the HH Blitz results for all members of the opsin family in human. There's actually 11 of them. And in addition to that, these PDAM sequences also map to some other GPCRs. And so the question here becomes, which of these PDAM hits are actually true opsin family members? To try to address this question, we've looked at a paper that published a chemogenomic analysis where they took sequence information in conjunction with ligand binding pocket information. And what you can see here on the right-hand side is a profile of 30 characteristic residues that are typical for opsin members. And so here you can see the Ballesteros-Weinstein numbering system, which shows that they are distributed over all the 7 transmembrane helices. When we now compare the residues in this ligand binding pocket for one of the PDAM homologs of human opsin protein, you can see that only 11 out of the 30 are actually identical. This raises the question, do we actually need all of the 30 amino acids? And so we've actually looked at a related question a number of years ago when we identified a minimal binding pocket for GPCRs based on the premise that GPCR function requires structural coupling from the ligands to the cytoplasmic domain. And so the ligand binding has to trigger conformational changes. And so in this approach, we are taking dynamics into account. You can see here is an overlay of the ligand binding residues, and you can see how they occupy a similar ligand binding pocket across all of the GPCRs that have been crystallized at that time. So the approach that we used is a method that Chris had developed previously called GREMLIN, which refers to generative regularized models of proteins and which had been developed previously for retrieval of information on long-range interactions from multiple sequence alignment. GREMLIN learns an undirected probabilistic graphical model known as a Markov random field, MRF. Unlike hidden Markov models, which are also graphical models, MRFs can actually model these long-range interactions as opposed to non-sequential, so as representatives of non-sequential residues. You can see this illustrated in a toy example here, how this is done. Column 2 is only occupied by a single amino acid tyrosine, which means that it's completely independent of all the rest of the multiple sequence alignment. So you can see that this node has no edges connecting into any others. In contrast, column 1 and column 4 are related. You can see whenever there's a serine in column 1, there's a histidine in column 4. And when there's a phenylalanine in column 1, there is a tryptophan in column 4. This means that these two co-evolved, and we can represent this by an edge connecting these two nodes. And so the overall idea is to analyze the entire sequence alignment and encode these long-range interactions via node and edge potentials. We've applied the GREMLIN model to the rhodopsin structure, and what you can see here is that many of the edges involve the retinal ligand. To verify whether or not this is true also for other GPCRs, we first looked at the null distribution for the GPCRs for which there was crystal structures available at the time, which you can show here on the x-axis with these numbers representing the ligand binding pocket size. And so the null distribution is this blue line here, which is going down as expected because the pocket is becoming smaller and smaller. In contrast, the GREMLIN edges a plateau here at around 50%, and only the last two receptors have lower values. But even for these receptors, the enrichment of edges over null is very strong and very statistically significant. And so what we asked is then, well, how small a binding pocket do you really need for a functional GPCR then? And what we found is that you actually only need 4 amino acids. And when we look at these 4 amino acids in the PDAM alignment, we find that 3 out of 4 are actually identical. This gives us very strong confidence that this protein is actually a functional G protein coupled receptor. The next question is, of course, is what about the specificity of the ligand? So in addition to being a functional GPCR, you also have to have the minimum requirement of binding that particular ligand. In the case of opsin, this is a lysine to which the ligand is covalently attached. And oftentimes, there are specific amino acids in the vicinity of that lysine, which is also referred to as the counter ion. This in rhodopsin is a glutamic acid, but you can see that our PDAM homolog has actually a tyrosine at this position. And so it turns out that this position very strongly affects the absorption maximum of the light-sensitive protein. And it turns out that there's also a tyrosine in the human OPN5, which suggests to us that OPN5 is actually the correct mapped PDAM protein as compared to bovine rhodopsin based on this analysis. So if you repeat this for all of the 11 human and 7 PDAM maps, we propose that there's actually only 4 PDAM opsins, those that are most closely related to OPN5, OPSG, OPSX, and OPSR. And this is particularly encouraging that we find red opsin in this list, because it's actually known that the corals don't only differentiate between dark and light, they can actually see colors. So they can actually, the larvae can actually preferentially attach to red surfaces as opposed to white surfaces. In future, of course, these predictions have to be experimentally validated. So this is a picture of my lab where you can see our biophysics spectroscopy setup on the left and a coral tank right next to it. In conclusions, I'd like to summarize that we developed a pipeline for membrane receptor identification in PDAM. We've told you a number of interesting stories related to opsin and vision and insulin signaling for nutrient concentration regulation. We have some other interesting stories for which I didn't have time to talk about today, relating to toll-like receptors in immunity, toxicity of plastics, as well as smell and taste. And last but not least, I'd like to point out that this pipeline is general. So you can apply this to non-membrane receptors, of course, but also to non-PDAM related proteins. With that, I would like to thank my collaborators on this NSF HDR synthetic coral project, especially Lenore Cohen, Noah Daniels, and Montserrat Olau-Sebikan for the homology research, Whitney Viscordes, Lokanda Kumar, and Nathan Brennis for the analysis, Holly Putnam, Jinkyu Yang, and Natasha Linsky for the coral biology and visualization. With that, I would like to thank you very much for your attention. Two tiny announcements. One is we are currently organizing an international art competition, which you are welcome to submit entries to. It's all about the comparison between humans and corals. And last but not least, we'd like to invite you to a special workshop on bioinformatics of corals at the upcoming PSB conference, which, as far as we know, is still going to take place in person in Hawaii. If not, it will be free if it's online. Abstract submission deadline is August 15th. Thank you very much.